Good morning, dear students. Welcome to Genesis. This is my fourth lecture on the chapter Absorption by Roots, and in this we are mainly going to cover plasmolysis, turgidity, flaccidity and role of turgidity in plants. Well, before we understand plasmolysis, let us just understand what can happen to a cell when it is kept in a hypotonic solution and in a hypertonic solution. Remember, if you remember in my last video, I had told that we only discuss the changes that we find in an animal cell, and I said in a plant cell, because there is a cell wall, the changes are going to be little different from what we see in an animal cell. So imagine a condition where you take a plant cell means it will have a cell wall, a cell membrane, and you keep this in a solution which is hypotonic. Hypotonic solution means the amount of solvent is more compared to the amount which is present in a cell. Now in a plant cell, nucleus is very conspicuous like in an animal cell, but another thing which is very important is a vacuole. So why did I mention a vacuole here? Because it plays a major role in the phenomena that we are going to study now. So here is the cell membrane and this is the cell wall of the plant cell, and here is the hypotonic solution where the plant cell has been kept. After some time, what we will see is, by the process of endoosmosis, since it's a hypotonic solution, water will start to enter inside the cell and will get stored in the cell. Where exactly it is stored? It is stored in the vacuole. So because of this, the vacuole is going to increase in volume, and as it increases in volume, it also pushes the cell membrane very close to the cell wall. So this gap that you can see will actually not remain if the cell is absolutely turgid because of inward movement of water. This condition where the cell wall is tight and rigid, the cell membrane is absolutely close to the cell wall, the vacuole has increased in size and volume, this condition is known as a turgid condition, which is seen in plant cells. Remember one thing, in a plant cell, because it has a cell wall, it never bursts due to endoosmosis, which can happen in an animal cell. So if you keep a red blood cell in a hypotonic solution, there are chances that it is going to burst if there is continuous endoosmosis happening and it keeps on increasing, the inner content keeps on increasing the pressure on the cell membrane, then it can burst. Such a phenomena usually we do not see in plants at the very beginning because the cell wall is very rigid and tight and it stops or prevents the cell from bursting. What will happen if I keep the same plant cell in a hypertonic solution? So in that case, if I keep the plant cell in a hypertonic solution, so this is the cell wall, here is the cell membrane, and I'll tell you why am I drawing it like this. You can very clearly see there is a gap between the cell wall and the cell membrane. Like we know that cell wall is freely permeable, so it actually does not stop anything from entering inside it, and it allows this solution to also enter. But this solution causes a problem. What is that problem? It causes the vacuole inside the cell to shrink and lose its water. So the water from the vacuole will go out from the cell into the environment which is outside. So this vacuole is now going to shrink, as a result of which the cell membrane, which was initially tight and very close to the cell wall, will also start to shrink away from the cell wall. So here is the cell membrane which kind of recedes away from the cell wall. This condition is known as flaccid condition of a plant cell. So I've shown you two conditions. When kept in a hypotonic solution, then the condition of the plant cell is known as turgid condition, and when it is kept in a hypertonic solution, that means the amount of water is less, the solute is more. In this case, the vacuole is going to lose water by the process of exoosmosis and the cell membrane will shrink away from the cell wall. This condition is known as flaccid condition. The flaccid condition is caused by the phenomena which is known as plasmolysis. So what is plasmolysis? It is the phenomena where the cell membrane shrinks away from the cell wall when a plant cell is kept in a hypertonic solution. Then what is flaccid? They sound quite similar. They are actually similar. Remember, plasmolysis is a phenomena and the state of the cell is called flaccid. That means when this phenomena plasmolysis happens, then the plant cell achieves the state which is known as flaccid state. So we discussed turgid state, which happens when you keep a plant cell in a hypotonic solution. Reverse of it is a flaccid state, which happens when you keep a plant cell in a hypertonic solution. And the phenomena which happens when you keep it in a hypertonic solution, where the cell membrane shrinks away from the cell wall, is known as plasmolysis. Achha, what will happen if I take this plasmolysed flaccid state of a cell which is in flaccid state, in a normal water? Like we pick it up from this hypertonic solution and now we bring it back to normal water. What is going to happen? The vacuole is again going to fill in water by the process of endoosmosis, and it is going to increase in volume. This is the condition which is known as deplasmolysis. So deplasmolysis is the condition which happens when you take out a plasmolyzed cell and keep it in normal water. Now, plasmolysis as a phenomena is very, very important for us, but because we kind of use this in our day-to-day life. When do we use this? We use this to kill bacterial cells by the process of plasmolysis, and this is done when you sprinkle salt on cut meat. If you sprinkle salt on meat, this excess of salt on the surface will not allow bacterial cells to survive. They will undergo the process of plasmolysis. Same concept is used while you are pickling. So when we pickle things, then the vegetables or the fruit, whichever is used in pickling, we put them in excess salt. Why? We ensure that there will be no bacterial growth, because bacterial cells are going to die by the phenomena of plasmolysis. This is also used if like for example, mostly by the gardeners, if you want to remove weeds from your garden or from your agricultural field. Weeds are unwanted crops that grow along with your main crop, right? So they kind of take away nourishment, sunlight, water from the main crop. So we do not desire that. So what do? We just sprinkle some salt on the roots or the base of the plant, and then the root cells undergo plasmolysis and the plant eventually dies. This can also happen when you are not aware about the quantity of fertilizer that you are supposed to put in your crop field. If you do not know, and if you overuse fertilizers, then these fertilizers will also cause plasmolysis of the root cells of your crop and will reduce the yield. Let us quickly practice a diagram to explain plasmolysis. This may come in your exam, or you may be even asked to label or identify the condition of the cell. So always remember, first and foremost, what you have to draw is a cell wall, and if you are trying to show plasmolysis, before that you have to show the condition of the cell when it has not undergone plasmolysis. So under such condition, the cell wall will be right next to the cell membrane, will be right next to the cell wall. So there will be no gap between the two, or very little gap. Then of course a nucleus and a big vacuole which is tight and filled with water. So this is the condition where, what are you trying to show? You are showing that the vacuole is tight, the cell membrane is absolutely extended towards the cell wall, there is no gap. Now the same cell, when it is kept in a hypertonic solution, what will it look like? So here the cell wall will remain as it is, but the cell membrane will recede away from the cell wall. Nucleus remains as it is, but the vacuole also decreases in size. So now you can see there is a gap. This gap is going to be filled by the hypertonic solution. This is cell wall, this is the cell membrane, here is the vacuole. So here you can see the content of the cell kind of shrinks when it is kept in a hypertonic solution. Now with this, there is one more diagram which you must practice, where we are going to show the different kind of pressures that develop inside a plant cell. See, in a plant cell things are little more complicated. It is because of the cell wall, but it is this cell wall which also protects the plant. Like I said, if you keep an animal cell in a hypotonic solution, soon it's going to burst, or it will shrink and it kind of loses its functionality. But that is not happening that quickly in a plant cell. So in a plant cell, when water molecules start to enter inside the cell, then the cell membrane, the vacuole, nucleus, they all develop a pressure within, and this pressure is exerted on the cell wall. So this is cell wall, vacuole, cell membrane. And what is this pressure called? This pressure, which is developed on the cell wall by the inner content of the cell, is known as turgor pressure. Turgor pressure. In return, the cell wall will also exert pressure on the cell membrane. This is how it maintains a balance and does not allow the cell to burst. That is known as wall pressure. And remember one thing, there will be development of osmotic pressure inside the cell due to continuous inward movement of water molecules. So there are total 3 different kinds of pressures which you must know to label along with the direction of the arrows. Be very careful with it, okay? So the turgor pressure is from within the cell towards the cell wall. The wall pressure is from the cell wall to the cell membrane, and the osmotic pressure is pressure which is created due to continuous inward flow of water into the plant cell. So now we will quickly discuss what is the importance of this turgor pressure or this turgid condition of a plant cell in a plant. Quickly understand how is this turgor pressure or the turgid condition in a plant cell helpful to a plant. The first benefit that a plant gets out of this is, number 1, it makes the soft tissues stiff. If you remember, I have already told plants do not have bones and muscles like we have. So for them to remain stiff, what they need is water. They have to have enough water in their vacuoles, which will keep the vacuole tight, which in turn will develop a turgor pressure, and this will help to remain, this will help all the tissues that are present, soft tissues that are present in the plant, to remain stiff, so the plant remains upright. During afternoon, you must have seen that when the rate of transpiration is higher in a plant because of the dryness in the atmosphere compared to the rate with which it absorbs water from the soil, this causes plant to lose or decrease its turgor pressure. Plant cells, they decrease their turgor pressure, and that is why the entire plant droops. But the same plant in the evening, when the rate of transpiration is less and the plant is absorbing enough water from the soil, again, as it regains its turgidity, the entire plant stands erect. Second application of turgor pressure is in seedlings. This basically helps seedlings, the roots of these seedlings, to push through hard ground. So this is the help that this turgid condition of the root cells provide to the plant. The seedlings can push through the hard ground because of the turgor pressure that is developed within the root hair cells, number 1. Number 2, sometimes this pressure is so high that it can even develop cracks in the walls as well as on concretes. The third very important function of this turgidity is it helps in ascent of sap. Let's understand what is ascent of sap. So ascent of sap helps in, ascent of sap is the upward movement of water in the stem. Now this is developed again because of turgor pressure, which develops another pressure in the root cells known as root pressure. What is root pressure? Root pressure is the pressure that is developed in the root cells due to inward movement of water by cell to cell osmosis. So this can be clearly understood. For example, if you cut a plant stem very close to its roots and attach a glass tube with the help of a rubber, then you will find water coming out of the stem into the glass tube, and the pressure which is developed by this flowing water can also bring changes in the level of the mercury in a manometer. So this is a very good example which shows that this pressure which is produced, which is known as root pressure in the root cell, due to its turgid condition, can push water upwards, because it can actually push the mercury level upwards. So this is ascent of sap. Ascent of sap is the upward movement of the water through the stem. The fourth application of this condition is opening and closing of stomata. So for this you have to understand the shape of the guard cells. We all know that the guard cells are kidney shaped, and they're little unique because the cell wall of the guard cell is thinner on the outer edge, but it is kind of little thicker as it is close to the stomata. So the cell wall which is close to the stomata or the stoma, they are more thick compared to the cell wall on towards the outer or the periphery. Now when these guard cells photosynthesize, because they have chloroplast, they will photosynthesize, they produce glucose. So when they produce glucose, then what happens? The concentration of these guard cells, I am talking about concentration of glucose, increases, and compared to the amount of water which is present in these. As a result of which, they tend to pull in water from the neighboring cells. Now when they start to pull in water from the neighboring state, they attain that condition of turgidity and they become tight. Now like I said, when they become turgid, since the cell wall here is thinner, so it distends more easily compared to the cell wall which is close to the opening. As a result of which, this opening is very clearly visible and this entire part remains tight and turgid. So it kind of pulls away the cell wall towards outside, so it becomes tight and turgid on the outer side compared to this area, and because of this you can see the opening, which is the stomata, which is required for various purposes, for exchange of gases, for exchange of water in the form of water vapor. So for opening and closing of stomata, this again is a very, very important concept in case of plants. So turgidity and turgor pressure, they are necessary for opening and closing of the stomata. And the last point application is the turgor movement. See, in plants usually we know that we do not see movement, but however there are some examples of plants which show clear movement, one of which is the Mimosa pudica plant, also known as touch me not. So these plants have leaflets, and when you touch these, any of the leaflet if you touch, you would see all the leaflets one after another start to close, and if you touch them little harder, then you will see the entire leaf, which has many leaflets altogether, will droop. What basically happens? What happens is, when you touch a small leaflet, they lose turgor pressure at the base of the leaflet. So here there is loss of turgor pressure. And there are many such leaflets which ultimately end up into a common petiole. So when you touch it little harder, the entire base of the petiole, this is the base of the petiole which is also known as pulvinus, it also loses turgor pressure, and as a result of which it droops. The same concept is also seen in plants which can move their direction as per the direction of the sun. Sunflower is a very good example. In all these examples that I have mentioned, it is because of these sensations, sensation of touch or sensation of light, they lose turgor pressure at a very important juncture, which causes movement in the plant. The movement can be drooping, the movement can be in the form of a rotation towards the direction of the sun. So today we discussed the benefits of turgidity in case of plant. Number one, it actually gives stiffness to the soft tissues of the plant. Number two, it helps the roots of a seedling to push through the soil, and sometimes it is so high, this pressure is so high that they can even cause cracks to appear on walls or even on concretes. Then it helps in ascent of sap, which is the upward movement of water through the stem. Fourth, they help in opening and closing of stomata, which is very essential in case of plants. And fifth, this helps in a phenomena called turgor movement, where plants show movement due to loss of turgor pressure. So we are going to meet again in my next video.